So I actually brought something evil, which is a PowerPoint, and I hope that I'll be forgiven for this, regardless. There we go. I actually have a PC, so there is no issue. It's one of these things. I think I wanted to be here today for a number of reasons. First, Boris was incredibly important in my life, and he was extraordinarily important for my desire to be a scientist, for my impetus to be a serious scientist as opposed to a less serious scientist. He was incredibly important for the feeling of power and cultural beauty in science that I think has impelled me through my career. And whether or not I've achieved anything that's worthy of Boris is another matter, but I think that the extraordinary inspiration that he provided was really, really very important. And I think the other reason that I'm here is that I just wanted to be able to talk about how broad Boris's reach was, because superficially you would say I don't work on anything related to what he was working on throughout his career, and yet in fact, in a deep structure of it, it's exactly what he was working on throughout his career. I was inspired in a very, very direct way by his teaching about gene and enzyme networks, about the interaction between transcriptional regulatory apparatus and the environmental signals. And I think more important than anything else, and the reason I have this title, is the sense that complexity and having many moving parts is actually no obstacle to understanding. In fact, it clarifies once you actually see the deep aesthetic structure that these networks are formed by. And so, and I think that in this sense, although I never worked in Boris's lab for more than a few weeks one summer, I think that he, in retrospect, had the greatest intellectual impact on my scientific life. So he was my teacher basically when I was an undergraduate at Harvard. He was a tutor, and I think Ed Skolnick will talk also about what that can do to one's mind, and he was unforgettable. And he encouraged me to try this MD PhD, and it turned out that I'm constitutionally unfit for an MD. I couldn't stand the hierarchy, and so that rapidly failed, but I ended up incredibly happily here at MIT as a graduate student. He overlapped, his department chairmanship overlapped with my graduate work at MIT, but actually, even though I was working in the cancer center for my graduate work, in a sense, his influence remained. And I think that the microbial physiology course, quote unquote, was an absolute electrifying intellectual event in my life. So just the way to think about the practice of science, the intertwining of science and culture, and this extraordinary way of conceptualizing the internal structures of complexity so that everything makes sense, and everyone's talked about this. So this is the complex system that I've chosen to try to push myself in this area, and this is hematopoietic cell development, and basically the set of mechanisms through which a multipotent precursor, just downstream of a stem cell, makes a choice about what kind of cell it's going to become. And I think many people who want to think about how this process is regulated immediately look for binary hierarchical choices. But if you actually look at the real biology, as Terry was saying, the real biology doesn't always follow what people want it to follow. And in fact, the ways you can get to many of these cell types do not go through single binary choices. They go through actually a kind of a valley crisscrossed with multiple paths. And so this is a challenge for us to think about. Does each cell type have its identity specified by a master regulator, a single transcription factor or a pair of transcription factors, perhaps, which masterfully regulates their genes? Well, there is indeed cell type specific gene expression that's driven by combinations of these regulators that are very well defined and very well characterized, but the same factor will participate in multiple different combinations, sometimes mutually exclusive ones. And although you can drive some of these programs by overexpression of some of these factors, in fact you have dose dependence, which says that the behavior of the factor is context dependent, and obviously this is not a pure master regulator paradigm of the kind beloved by mini review writers and editors of high-profile journals, but rather it's clearly a gene network issue. And in order to understand how these transcription factors are working together to create these very, very functionally important cell type programs, we have to understand more about their network relationships. Now, the system that I'm particularly interested in is T-cell development. And T-cells are made in a particularly neat, environmentally driven way, because these multipotent precursors migrate to the thymus, which then bombards them with ligands for the Notch signaling pathway, and that turns out to be the key environmental signal that pushes them through the whole process of acquiring a T-cell identity. And the thing about this is it's not a simple signal response, and in this sense it's quite different from metabolic regulation. In fact, the cells have to go through up to a dozen cell cycles under the influence of these signals. The onset of the T-cell specific genes is slow and cumulative and asynchronous. That is, it has a characteristic pathway, but there's no moment when the cell goes from not expressing these genes to expressing all of them. And furthermore, this process crosses over a period through which the cells gradually give up their access to alternative pathways. And really right up until this stage, which may be 10 cell divisions into this process of response to Notch signals, the cells still have the possibility, if you take the Notch signals away, of going off and doing something else. And only then do they resolve that they're going to become T cells. And so there are a lot of questions mechanistically about what kind of a regulatory system would behave this way. Why is the transition time so slow? Why does Notch regulate different target genes at different steps? And why are the steps irreversible? Given that Notch signals are needed throughout this process, what's really happening? So we've been interested in understanding this process, and it's been clear a priori that there were transcriptional regulatory changes occurring in response to the first wave of Notch signaling that would then change the context in which the next Notch signals were received, and then change the context in which the next ones were received. And from a lot of genomic work in my lab, we know what a lot of these transcription factors are. We know what their expression patterns are. And most important, we can see that they're both loss of expression and gain of expression changes that really coincide with this transition in which the cells become committed. And so we've decided to focus in on understanding really what is the push-pull in these cells. First, what are the cells doing when they give up their alternative cell fates? And second, what makes this process take a while? And it's very related to what we just heard, that this process is not what you might imagine inventing if you were going to design a way to make T cells yourself, but it is the method that is actually evolved to work in an organism like ours, which scales up, which requires proliferation of T cells precursors before they become committed, and you'll see why this makes a difference in a second. So this is we know that these are not just ways of adding more arrows to metabolic diagrams, but that at least 8 different transcription factors are needed for the cells even to get through this process, because if you knock them out, the cells don't stop very early. And so one of the questions is, what are they actually doing? Can you parse out the roles of the individual factors separately? And this became clear some years ago, and we were interested in trying to pull out or add or overexpress different ones of these inside and outside of the Notch signaling context in order to identify specific target genes. And so the idea is to make a mosaic of target genes for this whole pathway. And to cut a long story short, we ended up with a lot of interactions. These are not T cell genes. These are the progenitors, because we're trying to understand how the cells get from being progenitors over into this side of the diagram, where they're basically expressing T cell genes and shutting these off. But what we found very quickly was that we were missing a major categorical function that we needed to plug in order to get the system to behave properly. We were missing a negative regulator. And so we went back to this list of factors and asked, is there something that's turned on? And we indeed found what was turned on. It's a transcription factor called BCL11B, which is highly T cell specific. It's turned on exactly here. And we found out that it's the final rate-limiting mediator for T-cell commitment. So if you don't have it, the cells can't commit. They stay sort of eternally adolescent. Now, to understand how this, so the commitment process that's closed by BCL11B actually started way back here when the cell began to receive notch signals and turns on other T cell transcription factors. And so one of the big questions in the system is, what's taking so long? Why do the cells wait so long to turn on BCL11B? And it's important that they do, because BCL11B, if you overexpress it or express it prematurely, simply stops the cells from proliferating. And so we wanted to find the regulatory system, and this is sort of like the super sigma 54 regulatory system, because it turns out that the major, at least one major enhancer for the BCL11B gene lies actually about 800 kilobases downstream of the gene, in a gene desert that we found by doing epigenetic marking dynamics and looking at what regions actually undergo histone changes characteristic of de novo activation around the same time that BCL11B itself does. And this region then defines an enhancer that collaborates with the BCL11B promoter over a distance of almost a megabase in a very T cell specific way. And it turns out that by looking at the transcription factor binding sites at the promoter and at the enhancer, you can actually find a very comfortable kind of explanation for why BCL11B has such high activation requirements. It needs an input from notch signaling itself, but it also needs an input from two notch-dependent transcription factor target genes, TCF7 and GATA3, and others that were there beforehand. So we feel that we have some understanding then of the complexity of the pathway that results in finally turning on BCL11B at the right time. But this is just one part of the question. This is what turns on commitment, but what else, what is it fighting against? What's the purpose of the delay? And it turns out that the other participant in this is this factor PU.1, which is one of the regulators that was inherited early on, but then goes off during commitment. This factor is really famous for its ability to turn on genes of other types of cells. And so this is why the network has to include not only genes that are turned on to make the cell a T cell, but also what genes it is choosing not to express, because it's all coming into the thymus with the factors that would otherwise allow it to become something totally different. And if PU1 has its way, not only will it maintain expression of progenitor cell genes, but it will also try to drive the cell to become macrophage. However, and it will also block expression of a number of T cell genes in a very clear way. However, any amount of notch signaling actually tends to antagonize the ability of PU1 to turn on these macrophage genes. And what happens as the cells accumulate more and more experience of notch signals in the thymus is that then this now progressively makes this pathway more and more inaccessible, and it begins to protect these T cell genes from the repression by PU1. The result is that you now get activation of a positive feedback loop of these T cell transcription factors, GATA3 to MIB and TCF to GATA3. And the result is that this now also reduces PU1's ability to overcome the notch signal itself, so that this now allows the notch target genes and the genes that are regulated by other T cell transcription factors to collaborate to turn on the T cell program genes. Once this has arrived, and actually around the time that BCL11B is activated, now finally GATA3 and other T cell transcription factors acquire the ability to silence PU1 itself, and now PU1 is gone and these other alternatives are gone. And what we have is this one thing that I'd like to just point out is that during this period, when the macrophage gene activation by PU1 is blocked, nevertheless, the fact that PU1 is a transcription factor that can also regulate progenitor genes continues. And so what's lost when PU1 goes away is also the access to this reversion to progenitor status. Now, I've told you two fairly complicated things. But the point is that they actually make sense. And this is where I think this is where I feel that Boris is acting as a kind of a guiding spirit, and has been, while we were attempting to find some kind of sense out of this complexity. Because basically, what the cell is trying to do is not simply to make a T cell. It's trying to make enough T cells. It needs enough T cells for the body to be filled with T cells, but it, even more importantly, needs enough T cell precursors so that this very, very stringent selection that all T and B cells undergo in their development for having the right receptors for the body can actually be allowed. At the later stages of T cell development, something like 95 to 98% of every cohort of newly produced T cell precursors is chopped off because their T cell receptors aren't quite the right specificity. You have to be producing at least a 50-fold excess of T cells in order to be able to afford to use the immunological receptor rearrangement system that we have evolved to use. And so it's really important not only to make good working committed T cells, but to make enough of them. And so what the cells have to go through then is a succession of two different mutually antagonistic regulatory network states, both influenced by Notch signaling, but this one basically to maintain progenitorship and self-renewal potential, and this one basically to close the door on the other fates and to finish the T cell process. And I think that sense that out of all of those moving parts and out of all this complexity, the cells are actually trying to make sense for the body and for their program is one of the things that I've been hoping to emulate from the kinds of questions that Boris would ask me and would inspire me to ask. So my lab has been working on this at Caltech for a really embarrassingly long time. I think it's getting on to about 30 years now. I think that an acknowledgement for all the work that we've been doing is Boris's inspiration to look for that underlying order, and I think it's basically an aesthetic question, even though my diagrams have a long way to go to become aesthetic. But I think the people that did the work are, of course, here. But I think the most important thing really is just that I wanted to thank Boris for giving us the courage to take on really complicated problems, even if whether we solve them or not in an aesthetically satisfying way in my own lifetime is another matter. But the inspiration has been absolutely clear, and I am overwhelmingly grateful to this magnificent man for this, as well as for the many kindnesses and guidance that he gave me. So thank you.